Today I'm going to be talking about what protein misfolding is, what apoptosis is, and what it has to do with oxalates and Huntington's disease coming up. If you watched my last video, this is kind of following that information that I shared. It's just giving a little bit more information about why the oxalates are so damaging and why I really believe that it's the oxalates that are the biggest problem in our diet. Saying that insulin resistance and all of that stuff isn't important, because it is, but I'm saying that there are certain plants that we really have to avoid. As I've been saying over and over and over again, this is an autoimmune disease. This guy is one to watch for sure. He's definitely talking about protein misfolding, which is something that can lead to apoptosis. So that's where all the cells kind of clump together. You know, when the sick mitochondria clump together, all right, all the cells are clumped together and then they just die off in one huge clump. That's called apoptosis, and it's something that they've researched in Huntington's that can occur. And so that really fascinated me, because I found an article about kidney cell apoptosis, which in regards to I'm sorry, I want to say this right, oxalic acid, which is the acid from the oxalates. So, and I'm not even sure if I said it right. I'm so sorry about that. I am not a scientist. I'm not a nutritionist. I am just trying to investigate what works for me. I'm not saying anybody needs to follow my path. I'm just saying, I'm just putting out there my story, and take with it what you will, you know? And before we really get into it, let's go ahead and listen to this man's introduction, Ken A. Dill. He's amazing.
First thing I want to do is just introduce you, or remind you, for those of you who've seen this before, what a protein molecule is. They are very small. You have billions of trillions of them inside your body. And a metaphor, you could say, if you could scale up a protein molecule to the size of a penny, the number of protein molecules you have in your body is the same as the number of pennies that would fill the Pacific Ocean. You have lots of them. They're extremely small. Now, on the other hand, having said this, let me say also that protein molecules among molecules are very large and big and complicated, and this is a picture of one. This is a picture of a relatively small protein molecule. The beads that you see on this diagram are atoms, and the little lines connecting them are chemical bonds. And a key feature of protein molecules is that they are long and stringy, and they are made of, I've colored here, these are false colors, chemicals don't have color. I've colored these beads along the chain. They look like beads on a necklace. The beads themselves are the components of protein molecules, and they're called amino acids. Amino acids are very simple chemicals. You can go down to your drugstore, you buy powders. They're a couple of bucks. They go by names like valine and isoleucine and glutamine and phenylalanine and things like that. Those are the component pieces.
Remember, oxalates have oxalic acid in them. It's an acid. The same thing, okay, as what he's talking about here. That's why they can take over the same pathway.
This is a picture of showing you the string. There we go. This is just a schematic representation that strips out some of the amino acids so that you can see two things. One is the basic long stringy character of a protein molecule, and secondly, what an amino acid looks like along the chain. I've stripped out all the rest. Now, the first major development I want to tell you about is a thing called the protein folding problem and the protein folding code. This is has been considered one of the grandest challenge problems for the last 50 years in biochemistry. The protein molecule has a code, and the question is how to break it. And what I want to report to you is that a community of biophysicists has come now very close, has solved many of these problems, both high-level problems and the detailed problems, and we can now compute the structures of proteins in computers. Here's what the code is. Think about this necklace, if you like, as being made up of pearls that have 20 different colors. The 20 different colors represent the 20 different types of amino acids, so they represent the different chemical types. I'm just using a metaphor here. Suppose you could string those pearls together in any order you wanted to, red, green, blue, blue, pink, and so forth. It turns out the way you string those pearls together, the amino acid sequence, determines how this long stringy molecule crunches down into a crumpled version, which we call the shape. Now, one string of colors, blue, blue, pink, will crunch into one shape, and that shape may become a part of your muscle. A different string of pearl colors will crunch down into a different shape, and that will be, let's say, an antibody protein in your body. So the code is how the string of amino acids encodes what the shape is. That's the protein folding problem. I want to describe some advances in that area first. And then the second part of the problem is, once you know the shape of the protein, how can you tell what it does and how it works? These are the two key components for understanding the functioning in your body of protein molecules. Now I'm going to show you the folding problem and what we're looking to try to do. This is a protein that's all stretched out. You see 4 red colored amino acids here. And the basic idea is this is a chain that needs to find its shape. And we know that these 4 red beads, we know after the fact that these 4 red beads need to find each other. It's very much like you walking through a crowd of people trying to find your close friends. It's a fairly random process, but sooner or later you can sometimes do it. Those red beads need to find each other. This is called folding. And if you look at the blue strands, what they need to do is they need to line up like train tracks. And what the gold part needs to do is coil up like a helix. And this is showing you the folding process. And one thing you'll notice is that these pieces form, and they do it systematically. The other thing you will notice, so this is the final shape, and this is the shape that your protein has when it's in your body. This is the shape it needs to function. But the other thing you notice about this movie is it's very jiggly. Why is that? It's because protein molecules are so small that they get banged around by water molecules. Think about riding a bicycle in a windstorm. You're trying to get somewhere, but you keep getting blown around. And that's how protein molecules have to function too. They have to deal with these little microscopic windstorms. So this is the shape of a folded protein. The folding problem and the folding code is the business about, if I knew what the sequence of amino acids is, how do I figure out what the structure is? Now this next picture is showing you a more realistic, bigger protein molecule. Most protein molecules are bigger than the one I just showed you. They often look something like this. And now I want to switch from talking about the folding problem per se to talking about mechanisms and functions. And the case I want to make for you is that proteins are machines. You have 20,000 odd different types of machines in your body, and then other kinds of living organisms have other kinds of protein machines. There's tens of thousands to hundreds of thousands of different machines. And the first case I want to make for you is that these are real machines. That's not a metaphor. They use energy, they spin around, they pump, they act to cause force and motion. This is just a side view, a static view of a thing that is a protein motor. That light gray at the top is your cell membrane. Above it is the outside of the cell, below it is the inside of the cell. And this is just showing you a static picture, and now what I'm going to do is to show you a dynamical movie of how this machine actually works. This is a picture of that. This is a movie of that machine, and what you can see is it pumps acids, those are the little cubicle things, in and out of your cell so that it keeps the pH balance in your cell. And the things down at the bottom, the little cards going in and out, that's where the energy comes and goes. And what you can see is a ribbon diagram. So that shows you the nature of the protein molecule, and a critical point that I want to make about that movie and about how proteins function in general is they function by shape and by shape changes. And you could see that protein molecule going back and forth as different chemicals come and go. Shape is the critical thing here. Next, I want to show you another type of rotary motor. It's one that does not pump acids in and out of cells, but rather it actually creates force. If you're a bacterial cell, you need to propel yourself around in water to find food. What you need is something like the propeller on the back of a boat. But life doesn't have propellers for various reasons. Instead, what it does is it has long tails called flagella. And so now I'm going to show you a movie of a rotary motor that runs the tails on bacteria. Here you see it. Those are the tails. Now you're zooming in, and you're going to see the rotary motor in a kind of a cartoony-like fashion. This, too, is a real protein, and it uses energy, and it creates motion. That's rotary proteins. Those are a couple of many rotary proteins. Now I'm going to show you another kind of protein. This is a motor that slides back and forth. It walks like 2 feet. It's like a drunken sailor. Some of you who are old enough may remember those old cartoons about Keep On Truckin', where you walk like this. That's what these things do. The ATP and the ADP at the bottom where the feet are, incidentally they're called heads, but
I just want to stop it there for a second so that I can talk about ATP. ATP is the energy that we need to create in the mitochondria, and the way that we create ATP is through thiamine.
The energy comes in, and the motion happens in a general direction, but again, it's battered around by the water. And these kinds of motors are used in your muscle. So every time you do anything, it's sliding motions that are happening at the microscopic scale that cause that to happen. One of the great things about protein motors compared to real world, big world motors, if you like, is they don't make noise. And the last example I want to show you of a protein mechanism is this. Some proteins are valves and pumps. This is a case where this one too sits in a membrane, and it controls water flow in and out. Other such valves and pumps control lots of other things that go in and out of your muscles, and this is how your nervous system works, for example. If these kinds of proteins go bad, you get things like cystic fibrosis, for example. I've just shown you a few examples of tens of thousands of protein machines, but the case I want to make for you is just to convince you they are real machines, just at the super microscopic level. Now, one thing that's really interesting to those of us who are in this community about the mechanism of these machines is they don't work anything at all like big world machines. Let me tell you about 2 big world machines. One is electric motors, and the other is gasoline engines or internal combustion engines. We don't work by the principle of electric motors. Electric motors, you plug something in or you hook something up to a battery, and then the electricity causes an electromagnet to turn that into force. We don't have electromagnets. No living system has electromagnets. We don't plug in. We don't use batteries, except Iron Man maybe. Gasoline engines, they work on a different principle too. What they do is they take gasoline and they explode it and vaporize it, and then turning the liquid into a vapor applies pressure, and that's how it pushes on pistons and makes your car go. We don't have explosions either. We use a third principle. And the potential for this third principle is that we know it works because life has been around for 3 billion years, and so we've seen lots of machines that work this way, and yet we're not using it for any of our big world machines. So there's a lot of technological promise here that we could be using it for our big world machines. And the principle that I want to show you is just motion. It's just the motion. The protein is a blue blob here. The red thing is something that it's grabbing onto. This is part of its action. So proteins often bite onto smaller chemicals. This is how they often function. And now what happens is I'm going to show you that another molecule can come in and bind to the protein. Another chemical can come in and bind to the protein. And when it does, the shape changes, and then that causes the other molecule to unbind.
He trained at MIT, and he also trained at a bunch of different sciences, which is really fascinating. And so he says that the problem with the protein misfolding that leads to the cellular death, the apoptosis, is that the shapes aren't fitting in correctly because they're not the correct shapes. Okay? What's interesting about that to me is that when I watched it, I saw almost like a crystal trying to be put in the same spot, which reminded me of the prions from Huntington's disease. So if you watched my last video, you know that milk actually will shake oxalic acid off your joints and things like that. So what's great about that is milk does not misfold your proteins, okay? And so because of that, it's actually the perfect piece of the puzzle. So don't forget about that. Thank you so much for joining me today. I really hope that you learned something with this guy. He is amazing, isn't he? And we're gonna let him take us out because he's just got a little bit more to say that I think we really shouldn't miss. See you next time. Bye.
And then that causes the other molecule to unbind. This is the basic principle behind protein mechanisms in general. This is also the basic principle behind how pharmaceutical companies design drugs. Most drugs are designed against proteins. Let's say that blue thing is the protein inside a germ, a bad guy organism, some bacterium, something you want to kill if you're a pharmaceutical company. What you do is you then make a small chemical, the yellow thing, that goes in and binds to the bad guy protein. And so the bad guy protein drops whatever it was supposed to hold on to, and that kills the bad guy cell. This change of shape is the critical principle that works in biology. We know a little bit about it. There still remains to be a huge amount of science done.